All right, so now let's do a quick talk on reproduction and on meiosis. So let's start with some important terms. A gene is a short segment of DNA which codes for a single polypeptide or a protein. Each chromosome consists of a chain of many genes and some accompanying regulatory sequences and proteins. The genome is an organism's complete set of DNA. That includes all of its genes. Each genome contains all of the information needed to build and maintain that organism. In humans, the genome's about 3 billion DNA base pairs. The word haploid refers to cells that have only one copy of each kind of chromosome. Oftentimes you'll see it abbreviated as N, meaning one of each kind. In humans, the haploid number of chromosomes is 23. And the only haploid cells found in the human body are the gametes, either the sperm or the eggs. Diploid refers to cells that have two copies of each kind of chromosome. Diploid cells are sometimes referred to as 2N. In humans, the diploid number of chromosomes is 46. In all of the human body's normal body cells, the somatic cells, they're diploid. You might also see the word locus. That refers to the location of a gene on a particular chromosome. Where is the gene? Its location. So let's talk a bit about asexual reproduction. It involves only one parent. During the process, the parent produces clones which are genetically identical to itself. The benefits of asexual reproduction is that it can happen really fast. It's rapid, and it requires less energy than sexual reproduction, and only one organism is required to carry out the process. Some examples include binary fission in bacteria, budding in yeast and hydra, and the vegetative propagation that happens in some plants like strawberry plants. Please realize that plants normally reproduce sexually. This is sort of an exception to the rule. The major disadvantage of asexual reproduction is that it produces identical organisms. Since there's no genetic variation, the ability of the population to evolve is severely limited if they're all made by asexual reproduction. Asexual reproduction works well in stable environments to which the organism is well adapted, but it doesn't work well in changing environments in which populations need to evolve. Sexual reproduction involves the combining of DNA from two different parents to create an offspring. The major benefit is that it creates genetic diversity or genetic variation within the population, and this genetic variation allows the population to adapt and evolve. The major disadvantages are that two parents of opposite sexes are required. This is really nearly impossible for endangered species to be able to do. The process also takes much longer than asexual reproduction, and it requires a lot more energy than asexual reproduction. Most animals and plants reproduce sexually. Some protists and fungi also reproduce sexually as well. So now let's talk about meiosis. Meiosis is a specialized type of nuclear division that reduces the chromosome number in half. From each diploid germ cell, the cells that undergo meiosis, typically 4 haploid sex cells or gametes are created. Each of those gametes is genetically distinct from the germ cell that it came from. The two roles of meiosis are to create the haploid gametes, the sex cells, and to increase the amount of genetic diversity and variation found within those gametes. In multicellular animals, meiosis occurs only in the sex organs or the gonads. The only cells capable of doing meiosis are specialized diploid germ cells found in those gonads. Before meiosis gets started, the DNA of those germ cells is replicated during the S phase of interphase. Meiosis is often broken into two main parts, meiosis 1 and meiosis 2. We'll talk a little bit about those in the coming parts of this video. The very first stage of meiosis is prophase 1. During prophase 1, the events that happen in prophase of mitosis also happen. So the cell's chromatin, its DNA, it coils up, condenses to form chromosomes. They're going to be bivalent chromosomes, so they're going to look like X's. The nucleolus disappears, and the centrosomes form the spindle fibers, Also, the nuclear envelope begins to disintegrate. Chromosomes of the same type or same number, homologous chromosomes, pair up and they join together during a process called synapsis. So this is new. This doesn't happen in mitosis. This is unique to meiosis. The members of each pair contain the same genes in the same order, but they oftentimes contain different alleles or different versions of those genes. During synapsis or right after synapsis, the homologous chromosomes undergo this process called crossing over. And this is when they basically exchange certain segments of genes. So this process creates new chromosomes, genetically diverse chromosomes. It's a type of genetic recombination, and it allows parents to pass chromosomes onto their offspring that they themselves don't even actually possess in their normal body cells. It's a hugely important source of genetic variation. So here we can see prophase 1. So here we see synapsis happening between two sets of homologous chromosomes. Remember, in humans, there would be 23 sets because our haploid number is 23. Crossing over happens, and you can see how these blue chromosomes have a piece of yellow, and the yellow ones have a piece of blue because they swapped some segments. Again, the purpose of that is to create genetic variation or variability. Next here we can see some more crossing over, and you can see where gene segments from homologous chromosomes switch. Metaphase 1 happens next. During metaphase 1, the two members of each homologous pair, again, the chromosomes at this point are going to be bivalent, align across from each other along the metaphase plate, the middle of the cell. Each chromosome attaches to spindle fibers at a point on the centromere called the kinetochore. During the process of lining up, the paternal and the maternal members of each pair randomly line up across from their partner. The pattern in which each pair arranges itself is completely independent of the other homologous pairs. In humans, since there's 23 homologous sets of chromosomes, because of independent assortment, which this random alignment is called, the chromosomes can line up in 2 to the 23rd possible alignments. That's over 8 million different ways. And since these two rows of chromosomes will eventually end up in different gametes, this process of independent assortment alone gives humans the ability to create over 8 million genetically distinct types of gametes. So it's another important source of genetic variation. Here we see metaphase 1. And again, in humans, there would be 23 chromosomes on this side of the metaphase plate and 23 on this one. The colors here represent the parent these chromosomes originally came from. So remember that which side mom's chromosome lines up on each time is random. And that mom's chromosome from pair 1 might end up over here, or it might end up over here. From pair 2, it might end up over here, or over here. That's independent assortment. Next, we get anaphase 1. During anaphase 1, one member of each homologous pair of chromosomes is pulled toward each pole or each side of the cell by microtubules of the spindle apparatus. This process is called segregation or separation. The individual chromatids of each bivalent chromosome are not separated during this part of meiosis. Anaphase 1 separates the members of the homologous pairs from each other. That's it. So here we can see that happening. One member of each bivalent pair is pulled toward each side of the cell. Next, we get telophase 1. Telophase 1 is very much like telophase in mitosis. It's sort of the opposite of prophase. The homologous chromosome pairs reach the opposite sides or poles of the cells. The spindle fibers disappear. A new nuclear envelope forms around each set of chromosomes, and cytokinesis begins to happen, and it separates that original cell into two cells. The two new cells at this point are haploid. They have half the number of chromosomes as the original germ cell. But it is important to note that the chromosomes at this point are still bivalent. We count chromosomes by counting the number of centromeres. Remember, the centromere is the center part, so that has one centromere, even though eventually it will split into two separate chromosomes. So meiosis 1, let's summarize it. It's often referred to as reductive division because it halves the number of chromosomes found in the two cells that result. It begins with one diploid germ cell and ends with two haploid cells, which still possess bivalent or X-shaped chromosomes. After meiosis 1 is over, there's a period called interkinesis. It's like interphase, except it's very short, and there's no DNA replication that occurs during interkinesis. Then we go into meiosis 2. Meiosis 2 begins with prophase 2, and it is essentially just like prophase in mitosis. It happens to both of the cells that were created in meiosis 1. So the nuclear envelopes and the nucleoli of each of the cells disintegrate. The chromatin condenses into chromosomes. The centromeres replicate, or centrosomes rather, replicate, move toward opposite poles, and they grow. They construct spindle fibers. There's no synapsis during prophase 1. Prophase 2 rather. Prophase 2 is essentially identical to the prophase of mitosis. The same can be said for metaphase 2. It's essentially identical to metaphase in mitosis. The chromosomes in each of the two cells are pulled toward the middle where they line up single file along the metaphase plate. So in humans, there would be 23 X-shaped chromosomes in this line and 23 in this line. Anaphase 2 is almost just like anaphase of mitosis as well. The spindle fibers shorten and the bivalent chromosomes, which look like this, are pulled apart like this. This piece now, which was called a chromatid up here, is now referred to as a chromosome. And each of these now have a centromere at this point. So once the bivalent chromosomes separate, each half is now a full chromosome. These chromosomes now are pulled toward opposite sides. This set going this way, this set going this way, this set going this way, and this set going this way. Now, unlike mitosis, these sets may not be identical. Things like independent assortment and crossing over have mixed up the chromosomes so that we're not making cells that are identical to each other. After anaphase 2, we get telophase 2. Again, pretty much just like telophase of mitosis and the opposite of prophase. The chromosomes reach opposite sides of the cell. A new nuclear envelope forms around each set of DNA. The spindle fibers are broken down. The chromosomes unwind, become chromatin, and cytokinesis starts. It happens in both cells. You can see it happening there with the formation of a cleavage furrow. And when it's all over, we'll end up with 4 cells. These 4 cells have half the number of chromosomes as the original germ cell they started from. These cells will mature and they'll become gametes, either sperm or eggs. And these 4 are all genetically different from each other, and they're all genetically different from the cell they started with. So gametogenesis is the process which creates the gametes or the sex cells. It starts with meiosis, and that's followed up by a period of growth and maturation. The process of creating sperm is called spermatogenesis, and it begins with a diploid germ cell called a primary spermatocyte. Let's walk through that from a picture. So here's the germ cell. This has, in humans, this would have 46, at this point, bivalent chromosomes in it. Meiosis 1 happens. Remember, meiosis 1 is the reductive division stage of meiosis. It halves the chromosome number. So this cell splits into 2 cells. Each of these now are going to have 23 bivalent chromosomes inside them. Meiosis 2 now happens to both of these cells, pretty much the same way mitosis happens. Its job is to split the bivalent chromosomes in half. So these 4 cells that result from meiosis 2 are going to have 23 chromosomes, but they're all going to be made of a single chromatid. Those 23 cells, or those 4 cells actually, are going to mature and they're going to become functional sperm. Notice that all of these cells are haploid. So in humans, they would have 23 chromosomes, whereas the original spermatocyte or germ cell is diploid and would have 46 chromosomes. Now, this same process in females is called oogenesis. Some people say oogenesis. It's the creation of eggs. So here's the germ cell. This would have 46 bivalent chromosomes after S phase of interphase. It goes through meiosis 1. Notice that the cell splits in half, but it doesn't split evenly. It splits into one large cell and one little bitty cell. Both of those cells end up with 23 bivalent chromosomes in their nucleus. This small cell is called a polar body, and it's just going to die. In women, after meiosis 1, the process freezes, and it doesn't go any further unless that cell is penetrated by a sperm. If it is, the process of meiosis 2 is completed, where this cell is going to split again and create another polar body. This cell is going to have 23 single chromatid chromosomes in it. So is that one. All these polar bodies die. Their job is to essentially get rid of the extra genetic material so that the functional egg only ends up with 23 single-stranded chromosomes. After all that, the nucleus from the sperm and the nucleus from the egg combine during fertilization, and we end up with a diploid zygote, which will then do mitosis to become an adult organism. All right, let's talk about a couple of places where meiosis can go wrong. Non-disjunction is the main term. This is when homologous chromosomes or sister chromatids don't separate properly, typically during meiosis. So that can happen during meiosis 1. It can happen during meiosis 2. It could also theoretically happen during mitosis. It results in daughter cells with the wrong number of chromosomes. We call that aneuploidy. And that means not the right number of chromosomes. Not the right number of chromosomes. When non-disjunction occurs during meiosis, it results in gametes with the wrong number of chromosomes. And if these happen to get fertilized, by far the most common result is miscarriage. Most human embryos can't survive with the wrong number of chromosomes. But there are some conditions in which an aneuploid zygote can survive. It's typically going to have some serious consequences, though. Here we can see non-disjunction happening. So in this case, the problem happens in meiosis 1, where this pair of chromosomes doesn't separate correctly. So these cells end up with one extra chromosome, and these cells end up with one missing chromosome. Here, meiosis 1 happened correctly. The problem happens in meiosis 2, where this bivalent chromosome doesn't separate correctly. So all of these end up with the normal number of chromosomes, the haploid number. This one ends up with a missing chromosome. This one ends up with an extra chromosome. Some conditions caused by non-disjunction, probably the most famous one is Down syndrome. And this is where a child ends up with an extra chromosome 21. So they end up with 3 copies of chromosome 21 instead of 2. Another famous one is Klinefelter syndrome. These are males who end up with 2 X chromosomes and a Y. Turner syndrome is also pretty famous. This is a female that ends up with only 1 sex chromosome, only 1 X. This 0 here means there's not another sex chromosome, there's only 1. But remember, by far the most common outcome of non-disjunction is miscarriage or death of that zygote. So here's a karyotype, a picture of the chromosomes arranged in homologous pairs. This is a karyotype of a normal female. Notice there are 2. Again, we know this is a normal female because there are 2 copies of each pair of homologous chromosomes, as you can see. Pairs 1 through 22. Let's back up for again. 2 of each pair. Pairs 1 through 22 are the autosomes, the normal non-sex chromosomes. Pair 23, those are the sex chromosomes. And notice that the female has 2 X's, 2 identical ones. No Y's. So that's a normal female. Here's a karyotype for a male with Down syndrome. We know it's a male because in pair 23, there's a single X and a single Y. This person we know has Down syndrome because for all the pairs, there are 2 copies, except for pair 21, where we have 3. So this is Down syndrome or trisomy 21. Here's a karyotype for a female with Down syndrome. And that's labeled incorrectly. This is a female not with Down syndrome. This is a female with Turner syndrome. Because there are 2 of each pair of chromosomes all the way through until we get to the sex chromosomes. There's only 1 X. There are no Ys. So this is Turner syndrome. All right, that's going to be it for reproduction and meiosis. Hope that helps.